Okay, so this is the week 15 on our newly aligned schedule and the morning session for our review of the digestive system. And as always, this recording will be made available for you on the Canvas. There'll be a link to this, and then there will be also an evening session available at 8 p.m. So what I wanted to do really this week, we are going to focus on the digestive system. There's going to be a lot of anatomy that will focus. Of course, there's physiology involved, but I would say that overall, in terms of the digestive system, there's just so much to name and notice in terms of the structures. Other elements of today's objectives are fairly familiar or things that we can tackle a little bit quicker. In terms of the exam 3 and practical 3, which you finished just this weekend, I'm expecting to have those finished by the end of this week. So over the weekend, that's my time that I hope that I get the final changes done. I think that I have reviewed all of the answers couple of times by now, so I'll just do one more round of making sure. And as always, we'll have a bit of a then feedback as a class about common misconceptions, challenging questions, and some important concepts that I want to highlight of those. And of course, I have spent significant amount of time to get you also your individual comments. So once we get those released, I really encourage you to have a look of those as well. What I'm asking you to do this week is to cover the assignments that relate to the digestive system. And as you'll see, there's familiar elements such as the LearnSmart quiz and the anatomy practice or practical practice, if you wish. Those are all things that you know from the past. The only difference is that this week we actually have 2 labs. One lab on the lipids and one lab on the enzymes. So I would ask you to complete both of those, and there you can see the point accumulation as well, so you can look through those. Everything that we have assigned as work for this week is going to be due on the last day of this week, which is the Sunday, I believe, May 2nd. What I'm going to do now, I'm going to jump off from this presentation and we will go through the key points of the digestive system using a selected number of slides from our actual lecture videos. So let's do just that. My computer is running a little slow today, but I'm sure we can get this running in no time at all. And you should be in a few moments able to see the slides for the digestive system. So we'll use those to guide our discussion through some of the key concepts. But as always, I'm very encouraging of if you have any questions, just feel free to, if you're not present in this session, then send them to me. But if you're present in a session, we'll have time also to address those. You don't have to wait to the very end. So what we're going to do this in, I think, 3 parts. And the first part is really kind of getting the general basics grasp of those. So we'll have a look of key terms, movements, layers, functions of the digestive system. And you might remember this formula or series of reactions from our earlier discussions when we looked at how the respiratory system, urinary system, and cardiovascular system played role in transportation of this excess carbon dioxide and water that were produced as a result of the cellular respiration. So let's have a look of that. Hold on a second, as I'll get us to the right page. So now we should be back and everything running as planned. So we are familiar with this equation, which is the outline of the cellular respiration. What we see happening here is that we have sugar. In this example, we have one glucose molecule, and that gets changed into ATP, which was, of course, the chemical currency of energy in a cell. And making ATP out of these sugars is really all that your digestive system aims to do. That's the main goal. And all other steps are then steps to either achieve this or deal with the, for example, waste products or whatever we end up as part of this process. So this is the big picture that we want to do. We want to generate energy. And if we go back to our very basics, we can see how that works. But before we talk about that, I've listed a couple of functions of the digestive system. So obviously we do need to get the food inside your body. So taking the food in is going to be the first one that we have to consider. Of course, we also want to break these molecules, larger molecules, into smaller molecules. And you remember, whenever we broke large molecules into smaller molecules, as a result, energy was released. And it is this energy that we really try to harvest from the food that we consume. As a byproduct, we are, of course, left also with these smaller, simpler molecules or chemical components that then can be absorbed into the body as well, typically through the bloodstream. The exception to that was, for example, the large fat molecules, where we used the lymphatic system to transport those. But we'll come back to this important concept of breaking large macromolecules into smaller components and how that releases this energy that we saw here. And then we'll talk about this absorption a little bit more as well. Final thing that we do want to do, of course, with digestive system, what cannot be used, that needs to be also eliminated from the system. So that's just as important part as getting the material into them. The two terms that are going to be super important for us and that I kind of alluded to earlier on refer to digestion and absorption. So let's have a look of those. So when we talk about digestion, what we're really talking about is about the process of breaking larger molecules into smaller molecules. And we saw that on this side. This is what we want to accomplish, larger molecules into smaller molecules. And the reason why we need to accomplish that is that if it is a larger molecule, our body is not able to take it in. And by taking in, I mean crossing the membrane that separates the outside world and inside world of your body. If you think about it, the digestive tract is one long tube starting from the mouth, ending at the other end. And when material is inside this tube, it's still technically not inside your body because it has not crossed that membrane. So it's kind of a continuum of the outside inside you. So we need to break the molecules into smaller molecules, and that process is known as digestion. The process of these small molecules that we have produced during the process of digestion is, and then when we pass them into the body, that is known as absorption. So you cannot absorb large molecules. To absorb large molecules from food, you need to first do digestion. You need to break them into something smaller. that's a manageable size for your body to take in. So we can get these and take them wherever we desire to use them. And of course, as a result of this process, we are left with this energy that we saw. So those are going to be two important terms that I do really want you to be comfortable with, guaranteed to be asked in an exam typically, guaranteed to be the very foundation of what you need to know. So make sure that you're comfortable with the difference between digestion and absorption, with digestion being breaking large food molecules into smaller components, and absorption being then those smaller components crossing inside the body through the membranes. Then I have two different kinds of movements that I want to speak with you, and we've seen examples of this already earlier on. And this is kind of counterintuitive because in many schools and many textbooks, we actually cover this chapter before we cover some of the other material that you have seen. But we're following the order that your school uses. So this is why I'm introducing them at this point. So the first term that I want you to be comfortable and familiar with is the term peristalsis. What peristalsis refers to? It refers to the movement along the digestive tract or any other tract. We've seen examples of that, for example, in the urinary system. But this movement within this tract is not just random. It follows along the tube, squeezing one part after another in a synchronized manner so that it pushes food along this tube. So that's what generates the movement of the material to one direction and only to that direction where we want to go. And that's because of these muscle bands squeezing one after another, immediately one after another. All that pushes it forward. So the way to think of this, I often say that we have two kinds of people. And when you are in your relationships or visit someone, you're very quickly going to find out what kind of a person the person that you are and what kind of the person that you visit is, based on how the toothpaste tube is being squashed. So the people who work their toothpaste tube from the one end to distal end and squeeze it systematically so that everything comes out from the very end, where's the opening, and there's nothing left there. These people show an example of peristaltic movement in the way how they empty the toothpaste tube. Let's have a look at the second type of people that we have. Well, some people just grab the toothpaste tube from the middle and squeeze. Some toothpaste remains at the back, some comes out from the front where the opening is. This would be an example of the second type of movement that I wanted to talk with you, and that was known as segmentation. In segmentation, what happens is that there is a squeezing movement in a tube, but that's not synchronized at all. So it happens randomly at random places, and as a result, nothing really moves to one or another direction of a significant distance. It kind of stays within the same place, whatever is being squashed. Instead, segmentation is a great process for mixing materials and mechanically also breaking materials down as they get squished on this single point. But it doesn't really contribute to moving materials along this tract. So remember these two types of people when you are revising the concept of these movements that we see in the digestive system. Then I wanted to talk very quickly just about the digestive tract as a whole, and I've already referred to that, is that it starts from one end, from your mouth, ends to the other end, and all parts of the digestive tract other than a couple of accessory organs, and there's three that we will focus on at the very end of this session, but all other parts are a continuous tube. So continuous tube that never ends until it reaches from one end to another. It can get wider, it can get more narrow. It can have different kinds of membrane layers, linings, but really it is a continuous tube. And that's why you could consider that whatever has entered to the digestive tract still isn't technically inside your body until it's able to cross from there the membranes, so that it's inside the body in a more specific sense. So one thing that we notice with this continuous tube that we saw just earlier on is that it has a similar structure in all parts of it. The proportions of these elements that we'll discuss next might vary, but we end up finding these 4 layers in all parts of the digestive tract. And these 4 layers can sometimes be known as tunicas as well. If you think of what a tunica is, tunic that a female, for example, might wear, it's a piece of clothing that you put on top of another. It's a layer. So these are layers of the digestive tract. So don't get thrown off if you see sometimes someone referring to them as tunicas. The other thing that I want to highlight here before we start discussing too much further is that we're seeing a cross-section of the digestive tract and these layers on this view. And the very center of the tube is known as lumen. So whether it is a blood vessel, digestive tract vessel, or tube, or anything else, the center of a tube is always the lumen. So we're working and reviewing these, starting from the inside, making our way towards the outside. So we're working through them in this order. That doesn't mean that in an exam you could not be asked in an opposite order. So just don't let that trick you. Remember, the innermost layer is the one that we start with, but the order can be asked in reverse in the test. So we're going to start with the layer known as mucosa. Mucosa is the innermost layer of this digestive tract, general model that we see. And what we'll notice here, that the function of the mucosa is really to detect, secrete, and absorb materials. So as materials move along the tract, we want to make sure that the other layers underneath the mucosa are not going to be damaged if there's something coarse that's being, for example, moved along this tract. So that's why there's a protective layer of mucosa. In addition to that, we might sometimes need to put out some materials. So, for example, some secretions like stomach acids or so on from this layer or layers below. So it can play a role in secretion of materials as well. And of course, some materials get absorbed at certain parts of the digestive tract. So they get into this wall structure and then further on to the body through the mucosal layer. So mucosal layer really has these 3 functions that it needs to do. It needs to protect as materials move along the tract, it needs to secrete, and it needs to absorb materials. And in some parts of the digestive tract, mucosa is of course going to play a more important role than in other parts. Well, the next layer is known as sub-mucosa. And really the name tells you where it is going to be located. It is below the mucosa. And what we'll notice is that the sub-mucosa contains very often various different kinds of glands. So we just talked about materials being secreted into the lumen of the vessel or digestive tract. So many glands that contribute to this secretion through the mucosal layer are actually located within the submucosa. The other thing that we end up noticing in this submucosal layer is that we find a lot of blood vessels on this layer. So this is richly supplied with blood vessels. So that's something to kind of bear in mind when you're thinking of these different layers. And the final thing that I want to mention about the sub-mucosal layer is that we also find a lot of nerves within this layer. So if we think of having these glands, nerves, and blood vessels, why do we find those in sub-mucosa? Well, that's because these all contribute to the regulation of the digestion. So really, the large portion of regulatory functions is completed from this layer. The next layer that I want to talk about is the muscular layer. And you'll actually notice on this diagram that it almost looks like I have two layers there. And that is the case. We'll talk about that, why that is important. What we'll notice is that it's all smooth muscle. So we noticed smooth muscle was only seen in few places in the body. We remember that smooth muscle was seen surrounding the blood vessels, and now we're seeing it within the digestive tract. So smooth muscle is able to control the diameter of the blood vessel and also control the movements, diameter of the digestive tract. And I mentioned that it looks like there's two layers. So remember, this is a cross section that we're seeing here. The innermost layer, the first layer that we're seeing here, is circular. So if you think that the tube would be running here, it circles around the tube in this cross-section shape. So it's able to make the tube more narrow or wider. That's what the inner circular layer of the muscular layer of the digestive tract does. The outer layer is longitudinal. And what I mean by that is that, again, if we have this tube here, it runs along the length of this tube. So it's able to extend or shrink that overall length of this tube. It doesn't really control the diameter, it just controls the entire length. So those are the two layers that we end up seeing on the muscular layers of the digestive tract. There is a third one, which we see only within the stomach. And this layer is actually oblique, so it doesn't follow either one of these directions that we've seen before. It's something in between. And the reason why we find it only in the stomach is if we think of the importance of the function of the stomach is really there to mix materials. Mix the materials with the contents of the stomach, acids and so on, other secretions. So we want to have as many different kinds of movements as possible there within the stomach generated by the wall of stomach. So that's why we find this third oblique layer there. And the final layer that we're going to discuss in the general structure of the digestive tract is going to be serosa. Serosa is made of connective tissue, and its function really is, if we simplify it, to anchor the tube or the tract to wherever it needs to be held in place. So that's the main function of it. There are a few more things that I want to discuss in terms of the general concepts of the digestive tract. But at this point, what I want you to know is that we see those 4 layers, and they're present in all parts of the digestive tract. Their relationship to each other, their proportions might vary a little bit depending on what part, but at large, we do find them at all parts of the tract. So you can make a note of that. So the couple of other structures that I wanted to talk with you guys, first one of those is going to be our peritoneum. And you remember, peritoneum was this serous membrane that lines the abdominal cavity. We're seeing it here on this picture as this nice lining drawn with the red color. And there's a specific part of it that I want to highlight that becomes important. But before we jump to that part, let's think of why we would have peritoneum. Well, there's 2 functions that I want to emphasize. First of all, peritoneum covers all of these intestinal structures that are intraperitoneal. You remember we had some retroperitoneal structures which were behind, such as the kidneys that we talked about, but peritoneum generates these anchoring sites so that the organs remain at their right place. The other function that we actually see, which is, I think, very important and very noteworthy for you to make a note if you're into that, that peritoneum serves also for the purposes of generating compartments. And why compartments were important? We can refer back to, for example, when we talked about the heart. If we have a compartment where we're keeping that organ apart from the structures outside, that helps us to limit the spreading of an infection to other parts. So, for example, if there's an infection within the structures that are inside, so intraperitoneal, it wouldn't spread to the other parts of the body because it's enclosed within this structure. However, if there's an infection on the outside the peritoneum, it's not going to be able to enter to the intraperitoneal structures. So that's why generating these compartments is very important. Well, the two structures that I said that I wanted to, or few structures that I wanted to talk about in particular, the one that I want to highlight is known as greater omentum. So greater omentum is this fold here that we see. And greater omentum, the reason why it is an important structure for you to know if you end up doing any dissection work, if you end up doing any other work where you are entering to the abdominal cavity through from the front, so you'll notice that it forms almost this big flap-like appearance at the very anterior portion, fairly superficial to the abdominal cavity. So you will then either just cut it away or you might flip it upwards out of the way for other structures that you're looking at. But greater omentum becomes very important as you work through the materials. Before we jump off from this, I just wanted to remind ourselves about the importance of the referring to the parts of the body and abdominal cavity in particular in relation to the peritoneum. So you remember we had retroperitoneal structures. Those were located behind the peritoneum. So an example of that would be, for example, the kidneys, which we just had a look. Then we have intraperitoneal structures. Intraperitoneal structures are located within the peritoneal cavity. And finally, we have subperitoneal, that would be located below peritoneum. A good example of that would be, for example, the bladder. So bladder is located below the peritoneum, as we can see. So that's something that I just want to make sure that everyone's familiar with, how we use this structure as an anatomical landmark for our other discussions. What I next wanted to talk about a little bit is just going back to the basics of what you learned when you covered the nervous system. You remember that the autonomic nervous system had two branches, sympathetic and parasympathetic branches. And it is the parasympathetic branch that drives the activities of the digestive system. So when the parasympathetic branch is active, the digestive system works hard. So you might remember that this was sometimes described as this rest and digest branch, if that helps you to remember it. But this branch is not always active. The other branch that we had was, of course, the sympathetic branch of the autonomic nervous system, and that was activated in response to a stress. So that was often described as this fight or flight branch. And during the activation of the sympathetic nervous system, of course, our digestive system is inactive. So that's not going to be working as hard at all. So just that basic concept to remember. Finally, I wanted to highlight here with this diagram one thing that you might have already noticed when we covered the blood vessels is that there's a rich blood vessel supply within the abdominal cavity and especially to the intestines. Well, why would this happen and why would this be important then? That would be a good question. So when we have nutrients and energy that we are able to collect from the food that gets digested and absorbed to the bloodstream, this blood now, which is in rich supply within the digestive organs of the abdominal area, this blood is now rich in nutrients and energy. And one thing that we notice is that your body is not very good at planning ahead. So any tissues, if they get rich supply of nutrients and energy, they will just simply burn that off, that energy and those nutrients. They don't think about the future. So to avoid of all of this nutrients and energy that we have spent a lot of time and energy and resources of collecting, of being just wasted, we need to store it somewhere. And the structure that we're going to use storing majority of these is going to be our liver. And liver is kind of, if you want to described it in this way, a little bit more like the sensible one out of all these other organs and tissues where the blood would go. So liver stores the energy and the nutrients for whenever they are needed in the later parts of the individual's day or life. So after blood passes through the liver, then it goes to all other parts of the body. But pretty much all blood from the digestive system passes first through the liver. So that's very important. And we'll see then if you have liver damage, that can cause then issues that these energy and nutrients don't get properly stored. So liver cirrhosis is a good example of that. But we'll have a look at that a little bit more a little later. But at this point, I want to jump to actually the structures that we're going to discuss. And we're going to start by this question that where does the digestion really start? And if we try to be as specific of the body part where the digestion starts, that's a good challenge for us. So we remember digestion referred to breaking down these larger macromolecules so that the absorption, which meant the crossing of the membrane, can happen at the later part of the digestive tract. So where do we start digesting the food that we consume? Well, sometimes students answer to me that well, the first structure would be within the mouth, maybe one part, tongue, or other parts there. Some tell me that it's the teeth. Some go even further and say that no, it's the lips. Well, I actually say that it's even before the food enters to our body. Just a simple sight or smell of food triggers salivation. And our saliva contains many digestive enzymes, such as the salivary amylase, which breaks down the starches. So just the process of seeing or smelling the food triggers the digestive processes. And we are going to spend now a little bit time going through the oral cavity and important things that we need to notice from there. So first of all, what are the functions of the oral cavity? Well, the first one that we've already noticed is that that is the place where we take in the food. So food enters to our body and into this tract that was one continuous tube within through the oral cavity. The other important function that we end up noticing that the oral cavity has is, of course, the speech, its role in the generation of the speech. So our communication is largely verbal. So mouth and oral cavity is very important in that sense as well. And finally, the third one that I've listed here is the sensory reception. If you think of even the development of a child based on few different theories, child goes through an oral stage where child explores the world through the mouth. We also, if you think of the food that you consume, which was the first function that we listed here, well, we want to screen that that food is good to be eaten, that it hasn't gone off, it's not past its best time to be consumed, it's not going to make us sick. So all that screening of the food happens also within the oral cavity. So we have the taste, we have the texture, and of course then connecting to the nasal cavity, we are also smelling it. And we saw it as well. So we're checking it in many, many different ways that it is safe to consume that. Couple of terms that I wanted to highlight here that you might sometimes see being used if we use proper terminology. We can talk instead of about the oral cavity, or we can talk about the buccal cavity. And those terms, I just don't want you to get confused if you see those being used in this context. So don't let that throw you off. I think the other thing that I want to do at this stage is to review the borders of the mouth. So what different structures are giving us the various borders that we see for the mouth. And let's do that together, starting with the most anterior part. So anteriorly, it is the lips that form the boundary of the mouth, of the oral cavity. Laterally, it's going to be, of course, the cheeks. So on the sides, and on this picture, it's not that well visible, so you have to just remember that laterally, cheeks. If you didn't have cheeks, if you didn't have lips, food would just simply fall out. Let's think of the superior boundary. Well, that's going to be our palate. And we remember that we had a hard palate at the anterior portion and soft palate at the posterior portion. So at the front, we have bone within the roof of the mouth, and that's why it's hard. And at the back, it's only the soft tissue lining and uvula hanging from the very end. So let's look at the inferior boundary of the oral cavity. Well, that's going to be our tongue, this large muscle that we see here. And we'll talk about tongue a little bit more, but tongue is much larger than just the part that we see peeking out from the mouth. So keep that on mind. And posteriorly, so at the very back, we see our oropharynx, which then forms the back surface or back of the oral cavity. And we talked about the oropharynx already earlier on when we looked at the respiratory system, I believe. There's one more structure, one more name that I want to introduce in terms of when we talk about the different borders and different spaces of the oral cavity. That's going to be the vestibule. So vestibule, if you translate it literally, what does vestibule mean? It means a chamber. So the vestibule of the mouth is this space in between the teeth and gums, and then the lips and cheeks. This space is known as vestibule of the mouth. So that might be another term that you sometimes run into, and I want you to be familiar. Well, I mentioned that we're going to say few words about the tongue and why tongue is an important part. Of course, it formed the floor of the mouth. And what we notice is the tongue is actually composed of many different bundles, many different parts, but all of these are skeletal muscle. So the intrinsic muscles allow us to control the shape of the tongue, whereas the extrinsic muscles play more of a role in positioning the tongue. So tongue can be doing many different kinds of things in terms of gripping, repositioning, or mixing the food that we take in to the body through the oral cavity, through the mouth. The important thing that I do want you to know, and these terms will come up 3 times, so we have to be careful that we use a right term. Now that the food has entered into the mouth, it no longer is food. We talk about bolus. So when the food gets broken down and mixed with saliva, now we talk about bolus rather than piece of food that's within the system. So be comfortable with that term. There's going to be others that follow later on. You need to know in an exam that at what point these terms change and what they mean. Of course, tongue plays an important role in speech, in taste, screening the food, like we said, and it is the part that initiates the swallowing process by pushing the food back to the posterior surface of the oral cavity. A couple of other things that I want to say. We've got a lot of these small projections on the surface of the tongue. These are known as papillae, so be familiar with that term. And another structure that I think is definitely worth of knowing is frenulum. Frenulum is this small fold or ridge that you see made of connective tissue, both underneath your tongue, at the very anterior portion of your vestibule, from the gums to the lips, and then also at the top part, superior part, which doesn't show up on this picture. So frenulum are these little ridges, folds that we find within the oral cavity. And they will become important anatomical landmark later on. For example, when we describe or discuss about the saliva secretions, we use the frenulum to identify few parts that tell where the ducts open to the mouth. One more structure that I want to highlight, uvula. We've already mentioned that briefly a few times. So at the very back, coming down from the soft palate, we've got this thing hanging down projection. And what I have here, I think that we often talk so much about the typical average anatomy. I wanted to show you a little bit of a curiosity. What we see here is actually bifid uvula. It's divided in 2 parts. And this is something that happens during the development of the young individual doesn't follow the path that it should, and even prenatal or prenatal development. So the fusing doesn't completely get completed. You might end up seeing that as well. Next, I want to talk about the salivary glands. And salivary glands are, of course, going to be important for us since we noticed that it was the saliva that contained the salivary amylase, and that was the first digestive enzyme that we saw there. So saliva does many different things. First of all, it cleans the mouth, so we are constantly salivating, clearing away what shouldn't be there left anymore. That's what chewing gum bases on its function as well, that you start salivating and kind of cleaning the oral cavity as well. Of course, saliva is excellent in dissolving the chemicals, so that's something to notice. Also, if you did eat food and you didn't have any saliva, a lot of foods will be hard to swallow. Think of, for example, eating cornflakes with no milk, no liquid with them. That would be really painful to swallow if we didn't have saliva that gets mixed to it and that helps us to moisten the food and form this nice little rounded structure that's easier than to swallow. And you remember, this was now bolus, no longer food. Once we have mechanically started breaking, there's some chemical breaking, and it is mixed with saliva. Food turns into a bolus. And of course, we talked about the breakdown of starch by the salivary amylase as well, so the chemical digestion. Well, there's major extrinsic salivary glands and minor salivary glands. The minor ones are located within the oral cavity, whereas the major ones are located outside the oral cavity. And we're going to be focusing on these major ones. And I want to talk about 3 different types that we have. And we're going to start with these ones here. So the parotid salivary gland. Parotid salivary gland is located next to the ear, above the masseter muscle. The easiest way to remember that, at least that what I think of whenever I talk about parotid salivary gland, I think of a parrot sitting on a pirate's shoulder. So where the pirate would be is where the parotid salivary gland is located. So right next to the ear, above the masseter. What we notice also is that parotid salivary gland, since it's a gland located outside the mouth, it needs to have a duct. And this duct opens next to the second upper molar. So we're going to have a look of the dentition in a few moments' time. So that's going to be an important anatomical landmark for us to identify where this duct opens. Another thing to notice is that parotid salivary gland, a lot of nerves run through this area. So if we have extensive growth, for example, within the parotid salivary gland, and we would need to operate that would become quite challenging simply because of its location and what runs through this area. There would be a significant risk of paralyzing one of the facial nerves, for example. What I want to highlight before we move further is that the parotid salivary gland, the secretion from there is very serious, so very watery, that we secrete from there. The next one that I want to talk about is going to be our submandibular salivary gland. So submandibular salivary gland, located exactly where the name suggests, below the mandible. I would say that it's rather within the mandible, there inside at the medial part of the mandible, but it is a little low there, so that might be appropriate term to use. So submandibular salivary gland has as well ducts, and these open at the base of the lingual frenulum. You remember we talked about that earlier on, this connective tissue band. And in this case, it's the connective tissue band that's below the tongue. Right there, we see 2 openings for the submandibular salivary glands, ducts. And the thing that we want to notice then from the submandibular salivary gland is that the secretions from here are now going to be a mixture of serous and mucus secretions. And finally, we're going to complete this review by looking at the sublingual salivary gland, which, as the name suggests, is under the tongue. And this one is going to have multiple ducts, and these ducts open all to the floor of the mouth. So I guess that we're going to complete our review of the salivary glands by noting that the secretions from the sublingual salivary gland are now going to be mucus. So we're moving from very watery secretion at the parotid salivary gland to very mucus thick secretion at the sublingual salivary gland. And the submandibular is something in between the two. So the next question that I would probably propose is that how much do we actually make saliva in a day? And it turns out that that's about 1,500 milliliters, so 1.5 liters a day. That's a significant amount. And you could think of that, don't we then lose a lot of fluids? We just talked about that when we talked about the fluid balance after the urinary system and review of the urinary system. Well, this is not going to be an issue because most of the saliva we actually retain in the body by swallowing it. So it doesn't leave our body. Next, I want to talk a little bit about teeth, and I spent quite a few years working with training of the dentist. So this is something that I can get easily carried away with. So I'm going to try to keep this really simple and not get too carried. So teeth are, of course, some of the hardest structures in the body. And I want to talk about the primary teeth. We have 20 of those, which then get replaced by the permanent teeth, which are also known as our secondary teeth. So that's a term that you can sometimes see being used of them as well. And the pattern how the secondary teeth or the permanent teeth replace the primary teeth, milk teeth or baby teeth, that's actually very predictable. So we can use that to define the age of a young individual. Of course, when you have all the permanent teeth, that's not that good of a guide of aging an individual anymore. Okay, the next thing that I want to discuss quickly is the different teeth of the oral cavity. And what you see here is that this is actually easy. You know me, I like to keep things as simple as possible whenever possible. So you can simply divide the oral cavity into 4 sections, so quadrants. And we're going to go through one single section only, and the teeth within that one single section. And then you can mirror that to all of these remaining parts. And we're going to start from the midline, working our way towards the lateral side. So midline and anterior is the starting point where we start, and then we move our way to the back and side of the mouth. So the first ones that we're going to look at are incisors. So if you think why incisors developed, why would they be important, what would they serve, what sort of a function? Think of functions like biting an apple. These are ideally adapted, these kind of chisel-like structures that are really good at kind of biting into a material that's a little softer than some of the other ones that we'll look at later. The next ones are going to be our canines. So our canines are really kind of a result or outcome of our history, our evolution. They are adapted especially to tearing meats. So often very sharp. You might notice that Hollywood actresses and actors don't seem to have as sharp canines. Many get those actually sanded down by dentists, so they're not as present. But really naturally, we do have sharp canines that are ideal, especially for this function of tearing different kinds of things. The next ones that I want to talk about are our premolars, and premolars are actually sometimes known also as bicuspid. So if you think of them, they're something in between of what we just saw and what will come as the next teeth type. So they're not totally ideal for grinding, but they're somewhat okay for that purpose. And let's look at the grinding teeth as the last ones. These are, of course, our molars. So they're these large, flat surfaces that are ideal for grinding material between them with strong muscles, bringing them together and giving a chance to really get this material crushed and grind. So that's the dentition. I want to look at a general structure of a tooth just quickly. Like I said, I don't want to get too carried away in our review. So we're going to start the review of the tooth by looking at the part that's above the gum line. This part is known as the gum. That's the crown. So crown is all these structures that we'll look next that are located above the gum. So the first one that we see is going to be our enamel. That's the hardest material, one of the hardest, if not the hardest, in the body. So covers the outer layer of the tooth, and this is what we also want to protect. If we lose this layer, then we are more likely to get issues with the teeth getting decay and so on. The next layer is going to be the bulk of the teeth, which is our dentin. And following that, when we move further in, we have a central cavity where we find many blood vessels, nerves, and connective tissue that fills the central cavity. And it is especially when decay of a tooth reaches the central cavity that you get these extreme pains because there is this nerve supply. So if you need to get a root canal, this is where we try to get into and get those nerves there killed or desensitized so that the pain stops. Well, the second large part of the tooth that I do want to discuss just briefly is going to be the part of the tooth that's located below the gum line and within the jawbone. So this is going to be the root of the tooth, and we'll discuss that very briefly. Two structures that I want to highlight from there is the cementum. It does what the name would lead you to think. It cements the tooth into this socket that we have on this bony structure of the jaw. And then we have our periodontal ligament, which plays role as anchoring the tooth to this same socket as well. So that's the general review of the tooth. And I think that now that we have finished the level of the mouth, teeth, salivary glands, and so on, it's really good time for us to look at the other parts of the digestive tract. And we're going to also look at the accessory organs. So you can think of course, salivary glands as accessory organs as well. And we're going to start by discussing quickly about the pharynx. So you remember we talked earlier on about nasal, oral, and laryngopharynx. Well, in terms of the digestive tract, it's going to be only the oropharynx and laryngopharynx that we'll have a look. Of course, we don't have anything going to the nasal cavity if everything works well in terms of the food. So this is a pathway that's shared by the digestive and respiratory tract. And I think that what we need to notice from it is that they are made of we have two external skeletal muscle layers within the pharynx as well. The next thing that I want to talk about is going to be our esophagus. I find it confusing. In British English, you do put an O in front of the E. American spelling is a little different. What esophagus is, it's just a muscular tube that helps to carry the food from the end of the laryngopharynx to the stomach. And whenever the esophagus is not being used, it actually collapses or shrinks closed. So we don't keep that open at any other time than when the food is actually passing through. At all other times, you'll notice it's kind of squashed close. Then when food comes, it can open and extend. Couple of important things to notice. Esophagus, it's about 25 centimeters long and really has no digestive function. It is simply transport that it serves. And this transport, though, is going to be important in terms of the fact that it is a muscular tube. We can do now this peristaltic movement, which helped us to carry the food along the esophagus from the bottom of the laryngopharynx to the stomach. So it doesn't matter if you were standing on your head or if you were in zero gravity environment, such as in space, you will still have the food passing to the right direction because of this muscular movement of the material within the esophagus. Esophagus will pierce the diaphragm, so there's not many structures that will go through the diaphragm. This thin muscle sheet separating the abdominal and thoracic cavity. So we have our aorta coming through there. We have our esophagus, and we would have our inferior vena cava. But that's about it. Not a lot of structures can go through this muscle that we just discussed. And it's going to join the stomach then, and we're going to have a look of the processes that are involved there a little bit more in the next few slides. What we have here, remember now when the food had been mixed with the saliva and there had been mechanical breakdown already happening, we no longer talk about food, we talk about the bolus. So it is now mixed with saliva, broken apart mechanically, and also formed into this kind of a ball-like shaped structure that's easier to swallow and pass down these structures. What we have here is a microscopic image of the esophagus, and we can actually see the same layers that we just talked about in the general structure of the digestive tract. So we see our mucosa, the protective layer. It doesn't have any passage of the material in case of esophagus. Esophagus really doesn't do anything else than just transfer materials. There's no digestion, no absorption really taking place. Then we have our sub-mucosa, which was rich with vessels. You remember that? Then we have our muscular layers. Here we see two very clearly, and these were the ones that helped us to generate this wave-like movement known as peristalsis. And then we have the serosa layer, probably used more often a term adventitia, but rather than serosa, but we're talking of the same function, same idea. Okay, next I want to talk about the process of swallowing, and the fancy word that we're going to use for that, the proper medical term, is going to be deglutition. So what happens in swallowing is that first the palate, the soft palate and the uvula climb up, as we can see here. It seals off the nasal cavity, and epiglottis closes off the trachea. So we make sure that food is not going to go down to the windpipe, to the lungs. And then throat kind of jumps up, if you look, to catch the bolus. And then it just continues down the esophagus with these peristaltic waves. So the question that I could ask in an exam that would be kind of a tricky question would be that, is swallowing voluntary or involuntary? Well, the reason why it would be a tricky question is because first phases or first phase is voluntary. But once swallowing process has been initiated, it cannot be stopped. So it becomes involuntary at that point. So just something to bear in mind, kind of a good detailed question assessing how much detail has a student put into their studies. And on that note, I think that we're good to continue our discussion. And at this point, we reached the structure known as stomach. And stomach is this, if you want to describe it somehow, I would characterize it almost as a J-shaped structure. About 25 to 30 centimeters in length, just below the diaphragm, which would run here at the upper left part of the abdominal cavity. And what stomach is, it serves two functions. Of course, it is a storage tank for the food, so it serves as a temporary storage space, but also a lot of this chemical breakdown starts at the stomach. So a lot of secretions are put in there. They might not work quite as efficiently yet, but they are added to the content of the stomach, of the digestive system and contents of the stomach at this point, even though they might be activated later on. So let's have a look at the different parts of the stomach. But before we do, I want to highlight this concept that now that the bolus has reached the stomach and it is mixed with stomach acids and other secretions, we no longer talk about bolus. Now we start talking about chyme. And chyme is a little bit more liquidy content than what bolus is. So it's a mixture of a lot of digestive secretions and the food. And in that way, much more liquid in appearance. The volume of the stomach, of course, we do notice that can change drastically from empty to full. So stomach is really good responding to that, largely thanks to these structures that we see here, the rugae. Rugae are these mucosal folds within the stomach. So you see that they help us by making it small when it's empty and then extend when it's larger. So responding to that. But like I said, we're going to go through the different parts of the stomach. I want to spend a little bit of time on focusing on that. And we're going to start with the first part, where the esophagus that we saw coming down from here joins the stomach. That region is known as cardia of the stomach. So cardia of the stomach is where the esophagus joins the stomach per se. The next part that I want to highlight is the most superior portion of the stomach. This is known as fundus. And why fundus is important for us is that if you think it's above where the esophagus joined to the stomach at the cardia. So for this reason, a lot of the time, this plays important role when we're actually looking of medical images of the stomach and so on, because this is where the gases and air will build up if there's some within the stomach. So that's not uncommon to find air within the fundus of the stomach. The next part is the largest, most prominent part of the stomach. This is going to be our body. So majority of the stomach is the body. And final part that we see here is going to be the pylorus. Pylorus could be actually divided into two parts, the pyloric antrum and pyloric canal, but we're not going to go that far on this course. So pylorus is the last, most distal part of the stomach. And pylorus is going to be an important term. We'll talk about that a little bit more in few moments. What I do want to add to this diagram, which is very nice, of these 4 parts that we have learned at this point: the cardia, fundus, body, and pylorus. I'm going to also add a sphincter. This is going to be the lower esophageal sphincter. So this is muscle bands that seal off the esophagus from the stomach, right at the end of the esophagus. So that's why it's known as lower esophageal sphincter. And what's kind of cool about this is that if you do dissection, you can't really find it. It's not so much of an anatomical structure as it is a physiological structure. But the function of the lower esophageal sphincter is to keep this area closed so that the contents from the stomach don't end up climbing up back to the esophagus up there. And of course, what we know what happens if they do climb up there, you do get this sensation when the stomach acids get to the irritating to the esophagus. And this is, of course, known as our heartburn. Heartburn really doesn't have anything to do with your heart. It's simply the contents of the stomach irritating the esophagus, which is not designed to be dealing with this kind of acidic environment. What can cause then heartburn? There's many things: excess food, drink, obesity, pregnancy, running even. If you have prolonged failure of the lower esophageal sphincter and you have prolonged heartburns, it can eventually lead into esophageal ulcers. And if you keep irritating those esophageal ulcers, that increases the cancer risk on that side. So do get the heartburn under control if that's something that happens chronically to you. Well, there's going to be another sphincter. This is at the end of the stomach, so after pylorus, and not surprisingly known as pyloric sphincter. And this is actually something that we can see in anatomical investigation of this region as well. So also visible sphincter as such. And after that, we continue simply to the small intestines, which is something that we'll have a look in a little later at the next section. What I do want to say about the stomach at this point, I want to add a couple of parts or a couple of terms that we should know. So we have here lesser curvature, so it's not quite as prominent, and we have our greater curvature of the stomach. So some terms that can come in handy when we discuss processes and locations. Here, just as a kind of a clinical relevance, I've mentioned hernia. So hernia was whenever something leaves out from where it should be, something sneaks away from its normal location. And here we see part of the stomach squeezing through this opening that's meant in the diaphragm for the esophagus to pass through only. Some of it is sneaking through it to the superior portion, and that would be our hernia here to look at. There's many different types of hernias, and they can become an issue, but they can also be asymptomatic. The next thing that I want to discuss was these layers that we mentioned when we discussed about the layers of the digestive tract in general, when we talked about the muscular layers. We do see the inner circular and outer longitudinal layers here. But what's new, we now see also this oblique layer. An oblique layer was the third layer that gives more distension and mixing to the processes that take place within the stomach. Now that we've done quite a lot of anatomy, I think it's a good time for us to jump into physiological side of things. So we're going to talk about the stomach secretions. And what we'll notice is that the stomach is lined by the simple columnar epithelium. And within this simple columnar epithelium, we do find these gastric pits, which are kind of like these drops within the layer that we're looking at. And within there, we find gastric glands. And these gastric glands produce gastric juice as their secretion, and that's going to be about 3 liters per day that we end up producing. So let's have a look of different kinds of secretions that relate to the stomach. And we're going to start with pepsinogen that is secreted from the chief cells in an inactive form. So pepsinogen on its own doesn't really do anything. It needs to be activated. Well, it is the hydrochloric acid from the parietal cells. So our stomach acid that then activates the pepsinogen and makes it pepsin. So this is the active form of pepsinogen that's responsible for protein splitting. The next one that I want to mention is the gastric lipase, and you remember lipids, so it's involved with splitting the fats. And that's secreted again from the chief cells, but because we have now hydrochloric acid, the stomach acid present, the pH of this environment makes the gastric lipase very weak. So it's not working to its optimal ability. So we need to leave the stomach to small intestines where the pH normalizes a little bit more before the gastric lipase can start working on its job of splitting fats. I also want to mention the mucus, which is secreted from the goblet cells and mucus glands, and the function of the mucus is, of course, to protect the stomach wall. And finally, intrinsic factor, which plays a role in vitamin B12 absorption, secreted from our parietal cells. So you see that there's a lot of connections, so all of these are linked and play a role in each other. So I wanted to say again clinical relevance, and this time I picked the gastric ulcers. So these are sores that develop in the lining of the stomach, and this is caused by stomach acid damaging the lining. And there can be many reasons for that, such as having certain bacteria or taking too much aspirin and so on. What are the risks with that? Well, we do have the damage to the wall, inflammation and bleeding. This can also open a great route for bacteria for spreading. So these would be things that we want to be mindful. Let's have a look of the secretion of the gastric juices and the 3 phases that we find in the regulation. So the first cephalic phase or reflex phase, if you wish, is simply seeing, thinking, smelling, or tasting the food. The second stage, about 3 to 4 hours after the food has been consumed, our gastric phase. This phase is stimulated by the stomach wall being stretched, distended, and also by certain chemicals, such as proteins being present, coffee, low acids, that then cause gastrin to be released and hydrochloric acid to be released. The final stage, or within the gastric phase, you can also have inhibition. So that was the stimulation. You have inhibition, for example, if we have low pH or it's very acidic, or for example, in between the meals. And the final stage that I wanted to talk about is going to be our intestinal phase, where it is first stimulated and then inhibited. And the stimulation is by the food entering to the small intestines, and that encourages more production. And then inhibition by the wall of the small intestines, especially the duodenum being stretched and being acidity of the small intestines, and fatty content, which would then need more time to digest. So this keeps the amount of the material in the small intestines manageable and acts through reflexes. So I think that that's a good amount of detail that we need to know now about this regulation process. And I think the good question is why stomach cell, why it's not well adapted to absorption, really taking the material through these membranes. Why doesn't food really pass from stomach to inside the body at this stage yet? Well, this is because of two different things. First of all, there's a lot of acids within the stomach, so the environment is very acidic. So that prevents this absorption from happening. There is no villi in large content. So we need a lot of villi, a lot of surface area for good absorption to happen. And of course, we have this thick mucus layer covering the insides of the stomach as well, preventing the materials from passing. So actually, the main site of absorption is going to be the small intestines. So it's going to be important for us to pay attention and discuss the process within the small intestines. But I do want to say here, I kind of want to remind us of those terms that we were looking earlier on. So we consume food once it gets mixed with saliva and mechanically broken down. We now start talking about bolus. And once we reach the stomach and the secretions from the stomach, such as the hydrochloric acid, we now start talking about chyme. Chyme is much more liquidy than the bolus that we saw. And finally, we haven't discussed this yet, but when we talk about feces, this is at the end of the large intestines when the extra water has been reabsorbed. From the contents, and it's kind of drier. We don't lose a lot of the water through the feces. Okay, now we're going to focus on the small and large intestines, and we're also going to have a look of these accessory organs, mainly liver, gallbladder, and pancreas. So the small intestines start at the pyloric sphincter. You remember right after the stomach. And they end at the ileocecal open. That's the first part of the large intestines where the transition happens from ileum of the small intestines to the cecum of the large intestines. And this is really the main site of absorption. So it's important that you know that it is within this area where the absorption really happens. So the materials are able to cross this membrane and enter inside the body. Let's have a look of the small intestines. So we saw here the pyloric sphincter that we already discussed, and then the ileocecal sphincter is a little later. Parts of the small intestines. How do we remember the different parts of small intestines? There's only 3 to remember, so it should be fairly manageable. Well, think of a mnemonic of DJ ill. So a DJ, disc jockey, who's really sick in a way that he plays sick music, really good music. That gives us the different parts of the small intestines. So the first part, duodenum, is about 25 centimeters long. This curvature right after the stomach, the first part, not a super long part, like I said, and it curves around the pancreas, kind of nicely taking it into its armpit. That's the duodenum, the first part. The second part, jejunum, is about 2.5 meters long. There's really no transition that would be anatomically visible from jejunum and to the next part, which is ileum. So this is more of just a conceptual change from jejunum to ileum, or a change that's more of a physiological. Ileum is about 3.6 meters long, so very long, and joins the large intestines at the ileocecal valve, as I mentioned. So notice how long the small intestines is as a structure. And the other thing that we notice with the small intestines is that we have there a lot of these villi. So the surface area of small intestines is really, really large. So that's another thing to notice. So this large surface area, that's a result of the length of the small intestines as a whole, but also these villi, various folds, microvilli, that all facilitates that small intestines are ideal for absorption. So that if you spread out the surface of the small intestines, that covers about a size of a tennis court in terms of the surface area. So a large area, ideal for this absorption that we just saw. A few secretions that we end up finding within the small intestines. We have mucus from the goblet cells. And then we have peptidase, which breaks the peptides into amino acids. We have various sucrase, maltase, and lactase. So these break the disaccharides into monosaccharides. And finally, lipase, which breaks the fats into fatty acids and glycerols. And it is thanks to the pH of the small intestines being closer to neutral that a lot of these processes can now work more efficiently than what we saw with the stomach. That leads us to talk about the large intestines. And let's go through the large intestines as well at this point. And now we get to here where the small intestines ends and the first part of the large intestine start. And this first part is known as cecum. Let's change the pen there. So cecum joins to the small intestines, and the last part of the small intestines, which was known as ileum, giving an ileocecal sphincter, which separates the cecum and ileum from each other. So that was our cecum here. We, of course, see from cecum also our appendix hanging out. That's just a small structure that is comes out inferiorly from the cecum. We discussed this a little bit of earlier on, how it's an evolutionary remain most likely, and how it is susceptible for various blockages, and we might get an appendicitis. Yeah, so we might need to remove that as well. And there's a picture of this process there. The third part that we're going to spend a little bit more time talking about is going to be our colon. And colon can be divided into 4 major parts that we need to talk about. So in total, colon is about 5 foot long. And let's look at the different parts. So here we could see the appendix, and here the ileocecal sphincter as the small intestines ends and cecum starts. So now we see the first part of the colon is our ascending colon, about 20 centimeters in length. And then we have our transverse colon. Transverse colon crosses from the right side to the left side. And it's in a length of about 45 centimeters in total. And finally, we have our descending colon on our left side of the abdominal cavity, about 30 centimeters in long. and then sigmoid colon. It looks a little bit like it has an S shape, so that might help you to remember that, which is about 40 centimeters in length. And then we have, of course, our rectum, our anal canal, and our anus. About that, that's the last part of the large intestines. But really knowing these different parts of the colon is going to be useful. So just as a quick recap, in case anyone missed that, let me get a different color pen. So the first part, starting from here, after the cecum and appendix that we saw, we have our ascending colon, our transverse colon, our descending colon, and our sigmoid colon. Well, what does large intestines do then? This is the main site for reabsorption of the water. So not so much absorption happening there, but mainly the water reabsorption. And also some of the electrolytes will be reabsorbed there. We do have a great amount of intestinal flora that's beneficial for us, that lines the large intestines, and there's things like fermentation taking place, vitamin synthesis, and of course we control any pathogenic bacteria here. As a result, whatever is not taken back, cannot be used, we are left then with feces. And this brings us to start looking at these three important accessory organs that I promised. And we're going to wrap up with these this session. So let's start by looking at the liver. So liver is located at the upper right abdominal quadrant, just below the diaphragm, which would be located here. Notice that liver had a really rich blood supply, so from the intestines, so that's where the energy was stored. Otherwise, the body would just go crazy and burn off the energy from the energy-rich blood. The functions of the liver, there's a lot. There's a lot of metabolic functions, storage functions for glycogen, vitamins, filtering the blood, removing old red blood cells, detoxing alcohol and drugs. But the most important one for us is going to be the secretion of the bile. So bile is made at the liver. And that's something that often people get confused, and it's easy trick question in an exam. Remember, bile was made by liver, and gallbladder, then we'll talk about that later, is just to store and concentrate the bile. Gallbladder does not do bile, does not produce the bile. Couple of structures that we see there from the liver, but I want to focus on the ducts because those are going to be really useful for us to know. So the first duct leaving the liver that we see here highlighted is going to be the common hepatic duct. So common hepatic duct collects the bile from the liver and passes it down to this point that you can see here, from where it travels up to the gallbladder through the cystic duct. And then when gallbladder empties its contents, the bile, when it's needed for digestive processes, it comes back down through the cystic duct and then continues through the bile duct to here, this little opening, which is known as hepatopancreatic ampulla. So this opening is shared by the hepatic system and also the pancreas, which we see a duct coming out from there. So the same hole allows for both of these secretions to enter to the small intestines, and this was the duodenum of the small intestines that it opened to. Next, let's have a look of the gallbladder. And before we jump there, I just want to mention about the liver. Liver has a lot of functions. I once really put myself on the test and tried to list all the functions. I think I got about 200 functions and then gave up and decided that I have other things to do in life than list. So liver serves many functions, but for our purposes, remember the function as production of the bile. Let's talk a little bit about the gallbladder. Gallbladder, you'll find it at the ventral surface of the liver. It's this thin-walled muscular sac, and function was the storage and concentration of the bile. So bile gets stored there until it is needed. Bile release is through the muscular contractions, through the cystic duct and bile duct, as we saw. Sometimes we can end up with gallstones. So these are caused by having too much cholesterol or too bile salts. That's really, if you want to think of it, it is this cholesterol escaping and escaping the bile and crystallizing in there. And that can become a problem, and there's many different ways of dealing with it depending on the severity. And the final one that I want to mention a few words about is going to be our pancreas. So pancreas is an interesting one because it has both exocrine and endocrine function. And we talked about the endocrine function when we discussed the hormones and everything going on there. That was, of course, the production of the insulin and glucagon. But the exocrine function is what we're going to be interested on this chapter. So that's the production of the pancreatic juice. And pancreatic juice is actually the most complete digestive secretion of the system. So this is the one that does a really good job in digestion and helps with absorption through that as well. And that was the one that was secreted to the duodenum through the pancreatic duct at the opening of the hepatopancreatic ampulla. So that's something that you want to make sure that you do recognize the pancreatic juice as the most complete digestive secretion of these systems. Here we see pancreas, the location really, it is retroperitoneal, located at the greater curvature of the stomach, and then duodenum wrapped around it, as we saw here. And I think that that's a good place and good level of detail for us on this chapter. And I'm going to jump off from this presentation. And we're going to go back to our initial presentation. So bear with me as I get that open for us. And in just a moment, you should be able to see again the presentation where we started. And we're going to jump here to the digestive system key concepts. A couple of things that I want to still mention before we wrap up this session. Some students at this point of the semester start to face that their Connect access code might be expiring. If this happens to you, contact me and we'll figure out a way. I probably wouldn't ask you to buy another Connect access code this late in the semester, but if you can, try to make sure that it doesn't expire. If you have any missing assignments, also goes for the exams and practicals, I am willing to work with you. We can figure that out. Just message me directly with the specific details of what assignment is involved, and we'll figure out a way that you can get those points. We can do it together. And finally, at this point, students often start asking me about the finals week. It is a chance to gain extra credit. So I have a little project that you can complete. It's worth of doing. It's only positive extra points towards your overall grade. And I will publish details of that soon. But it is really an independent project where you look into something of your choice. It can be a clinical condition or a topic that we're covered, or even systems interactions. And you prepare some kind of a thing about that. Most students prefer to do a poster. I'll give you a template, electronic poster. You can, of course, do a presentation, essay, or anything else that you can think of, even interpretive dance, if that's your way of presenting information. So I'll provide more details. It really doesn't take a huge amount of time. And as I said, it's only extra credit. So I would really recommend that you don't hesitate to tackle it and you at least give it a go. So that's really taking us to the point that I want to remind you again about the tutoring available through the Cranium Cafe. And if you have any questions, do contact me. I should be fairly well available. I know I have few messages on Canvas, and I'll try to get those addressed in just a moment. So that's really all that I had for today's session. And we will have one more session this evening. But if you're joining me through a recording or anything else that I can help, don't hesitate to message me. I thank you for joining me, and let's make it a great week. We're almost at the finish point. Bye.